Thank you so much. I'm really delighted to be here. It's a real pleasure to be able to talk to you tonight about something I'm rather passionate about. It's not the study that I did at university. It's something I had the opportunity to teach because there was so much general interest in this. And it was an opportunity to reach an audience of students who were interested in astronomy, but a little bit more specifically in the search for life in the universe. And it was a way to teach them science without them really taking a course in physics or chemistry. It was a way for them to learn their science through a topic which was inherently interesting to them. So let me dive right in. As Michael mentioned, there's a very brief introduction here. I went to the State University of New York in Stony Brook. I am a New Yorker, if you, give the dog a cup of water. You don't like coffee. So I am a New Yorker, but I did my PhD at the Ohio State University, but also in conjunction at the Space Telescope Science Institute in Baltimore. And this is where I met one of my thesis advisors, Keith Horn. The Space Telescope Science Institute is the institute run by NASA that runs the Hubble Space Telescope. So I was extremely fortunate to be there at the time. After I got my PhD, I went to Keele University, the other Keele University, the one in England, not the one in Germany. And then I went to the University of Texas, which being a New Yorker was almost as strange as going to England. It was a foreign world to me. But then after that, I went to San Diego State University, where I've been there since the year 2000. Since I've been there, the most significant thing in my career was being selected to join NASA's Kepler mission team, part of their science team in 2007, 2008 is when we really started. And that has greatly influenced the trajectory of my career. Unfortunately, the mission has ended, but it was a great run for about a decade, really. So let me mention very quickly, if you were to ask me what's the most significant thing I've done in my research, I can honestly say pretty easily, it was being part of a team that helped discover a type of planet that orbits two stars, so circumbinary planets. So our team was responsible for finding the first ones, and we had tremendous success doing this once we got started. It was very difficult. These are planets with two suns in their sky. Right? So you can think about, the Star Wars Tatooine, and it would not be too far-fetched. The planets we found are all gaseous planets. They don't have, solid surfaces. But, we'll get there. We're not there yet. So I was super fortunate to be part of this research and get a lot of publicity and high-profile coverage in news media and stuff like that, which was nice and fun. But the real work is doing the science. And as of today, we actually now have 17 of these planets, which is a very small number compared to all the planets we have out there. But these are very rich and wonderful worlds. One of them is one of the most fascinating worlds I can think about because it has 3 planets orbiting 2 stars. So the little video down there shows you the idea of what's going on. The planets orbit the stars. They don't go in between the stars or figure eight or anything like that. That's dynamically unstable. So with that, it's enough about me. Let's talk about astrobiology. So what is astrobiology? Literally, it's the search for life, or the search for understanding life in the universe. And when I was an undergraduate student, 100 years ago, probably, I took a course on this topic. And it was a very good course, and the lecturer was magnificent. But looking back on it, there was very little to say. It was a course about astronomy, and it was very light on the biology part, because there really wasn't a whole lot to say. And it was pretty light on the astronomy part, too, because, well, all we had was the solar system, and there was interest in Mars. And Mars was probably the only topic that people really paid much attention to. The point of tonight's talk, though, is to mention 4 topics which I personally think are remarkably important and relevant discoveries. And this changes the field of astrobiology from something that's speculation. People have been thinking about this for millennia, but the scientific investigation really is about 100 years old. But not a whole lot has happened besides, hypothesizing and guessing. Now things are testable, and now things are getting really interesting. And the reason for that is, I think, due in part to these 4 discoveries. So in tonight's lecture, I'll go through these 4 things. It should take about 3 or 4 hours. I hope you're... No, just kidding. Okay, so let's, before we go there, let me just back up. Exoplanets is my field of specialty. So I'm going to spend very little time on this. You can learn about exoplanets. It's a very hot topic in astronomy. The Nobel Prize was awarded just a few years ago for exoplanets. So I won't dwell too much on this. I'm happy to answer any questions you have, but it won't be the focus. But of course, first and foremost, I will talk about it. It's the next 3 I really want to get to. So let's start off with just the history of planets in general. So for all of humankind, since humans first walk on the Earth, probably somewhere in Namibia, we knew of 5 planets. And that was it, right? 5 planets. The most important discovery of any planet ever occurred around the year 1600, 1610, and that was the discovery of planet Earth. Now, this sounds kind of silly, but if you don't know that Earth is a planet, you're in big trouble, right? You're in big trouble. Your understanding of the cosmos is very, very limited if you don't know the Earth is a planet. So that is the most significant discovery, and it probably will remain that way. Let us fast forward to 1930, and then we have 9 planets, if you want to include Pluto, and we will not go there and discuss that. We'll just move on. 1995 is when things get exciting. This is the discovery of the first planet orbiting a star that is not the sun. So we call these exoplanets or extrasolar planets. Extrasolar was the first name, and exoplanet is much shorter, so we just use exoplanet. So the first one discovered in 1995, Michel Mayor and Didier Queloz, the Swiss team, won the Nobel Prize for this groundbreaking, fantastic work. So that was the picture in 1995. What is it today? It's astounding that we have 5,690 confirmed planets as of today. That number will be higher next month. And this is due to these ongoing missions and surveys and projects that are finding all kinds of new planets on a weekly basis. So this is part of the big deal of why we're talking about astrobiology now. We're not talking about just Mars. There's thousands and thousands of planets out there that we have found. And our technology is pretty limited. So the actual number of stars that have planets is estimated to be in the hundreds of billions. I'll get to that in a second. So as Michael mentioned, I was part of the Kepler mission. And that was a wonderful, wonderful experience. To just summarize what Kepler has done now, it has responsible for discovering about half of all the known exoplanets, 2,773 of them, plus about 2,000 more, which I didn't include in the tally. These are candidate planets, meaning they're probably planets, but we're not really sure. We're sure at about the 90% level. So probably of those 2,000, 1,500 might be real. The other ones are a little, suspicious. There might be noise or some other glitch in the software, or in the hardware of the telescope. But, putting it all together, this is a lot of new planets. And significantly for this topic, there are 361 that are in the habitable zone. The habitable zone is the region around the star where if you put a planet like the Earth, it would be able to have liquid water on its surface. The habitable zone does not mean anything about life, doesn't mean there's anything there, doesn't mean there's water there. It just means you could have water if you had a rocky planet, if you had a rocky terrestrial planet and you put it in the habitable zone, water would stay in the liquid state. If it was further out from the star, it would be too cold, it would be ice. If it was closer in, it'd be too hot, it becomes steam. So habitable zone planets are important because they have the potential of having liquid water on their surface. But just because a planet's in the habitable zone doesn't mean anything, right? It just means it has the potential. I'll give you a great example. There is an object in our solar system, which is terrestrial, pretty good size. It's exactly at the right spot in the habitable zone, and it is completely lifeless and dead. And that's the Earth's moon. It's right in the habitable zone, and there's not a drop of water on it. Not a drop of liquid water. Okay. Of those 361 planets, 23 have been measured to be about the size of the Earth. So if you have that planet in a habitable zone, it's this gigantic planet, 10,000 kilometers across. It's not a very interesting place because it's going to be covered in gas and there's no surface, and it won't be like Earth at all. But these 23 are like the Earth. Now, we can push through and do the statistics and do the estimates here, and you don't worry about what type G and K stars are. They're small stars. Stars like the sun are G stars. M stars are even smaller. But the point is, that number, 25% or 50%, is a big number. It's not 0.0001%. It's 50% of small stars have planets in a habitable zone. Whoa, that's a big, big number. Okay, so if you crank the numbers a little bit, and you realize M stars are the most common stars in our galaxy, and just because you have a star doesn't mean you have just one planet. You could have 2, 3, 4, 5, 6, 7, 8, 9. So these stars have multiple planets around them. So when you count this and you do the math, you probably have more planets than stars in the galaxy. That number is a few 100 billion. So that's why things have changed. That's why the first topic we spoke about is exoplanets. We now have hundreds of billions of worlds to explore. Now, in 2016 was an amazing discovery. Not the most monumental thing that you'll find on the front of the New York Times or something like that, or one of these big journals. But I think it's really important because of its almost philosophical significance. A habitable zone planet was found orbiting the star Proxima Centauri. On this figure here, it's towards the left, there's a little box near Alpha Centauri there. That's Proxima. Proxima is a very faint star, but it is the closest star to the sun. So if you ask the question, let's go exploring the galaxy. We'll go from star to star, moving our way outwards from the sun. How many systems do we have to explore before we find a habitable planet? One. The very first star you come to has a planet in a habitable zone. What this means is habitable zone planets are so incredibly common, they're not rare. The first star you come to, it's there, okay. So it means we have an enormous haystack in which to search for our needle, but there's hundreds of billions of needles, so it's not that hard. You point to a star in the sky, and unless it's a particularly bright star, chances are it has planets around it. It's an interesting thought. Okay, now let's get on to the more meaty part of my talk. So, extremophiles. Extremophiles are forms of life, usually bacteria or something called archaea, which are very similar to bacteria. Okay. Okay, so this is the people who are working on archaea. They're the ghosts, because they say archaea are not bacteria. Archaea are not bacteria. They're a third form of life. They are not. It's true. There are 3 forms of life on Earth: the archaea, the bacteria, and the eukaryotes. You can think of them as bacteria, but they're not. But they look like bacteria. Anyway, most extremophiles are archaea or bacteria. These are organisms that thrive in extreme conditions. Extreme, according to our point of view. Things that we can't imagine life could survive in there. Imagine, there's a geologic vat, a geologic pool out there, and it's boiling sulfuric acid. It's like, oh, nothing lives in there. Oh, there's a whole bunch of stuff living in there. There was a case where in the US, because of the Cold War, they had stockpiled food and put them in underground nuclear fallout shelters. They irradiated the food with gamma rays to make it sterile so it should last thousands of years. Decade later, they go down there, the food's all spoiled. So what? These bacteria don't care about gamma radiation. They're like, yeah, bring it on. It's just food. So the bacteria completely survived. The food cans are all bulging and spoiled. And this is one of the ways we discovered these bacteria that can withstand crazy high doses of radiation. Radiation sterilizes most things, but not all. Okay. Sorry about that. I see what's happening. There's a button that shuts off the screen. Don't push that. Okay, so let me just talk very quickly about some of the forms of life in the extremophiles. So heat. Heat is something we use to sterilize things. If you bring it to boil, it should sterilize it. Well, that's not true. There are some species that thrive at temperatures near the boiling point. And that's one of the distinguishing features about extremophiles. They require extreme environments. They don't just withstand it. They're not just tough. They need this crazy high environment, this crazy harsh environment, or they die. An extremophile, if you try to grow it at room temperature, normal conditions, it would die. So Pyrolumus fulmari wants to be at the boiling point of water. If you bring it to room temperature, it freezes to death. It's too cold. Its proteins don't operate. The record holder is this wonderful science fiction name, Strain 121. It survives at up to 130 degrees. That's crazy hot, and it survives there. It can tolerate that heat. It doesn't like it. It preferred if it was only 120 degrees. It can reproduce and thrive in that environment. At 130, it says, this is a bit warm for me. But it is the record holder. So we got heat, we can have incredible pressure at the bottom of the ocean. There's so much water above you at the bottom of the ocean, the pressure is so high, most things would get squished into a jelly instantly. But not these extremophiles, they're happy down there just surviving. Salt, you can have incredibly salty environments. It's kind of ironic that people who are on the ocean and in rafts can die of dehydration, even though they're on the ocean, right? They're surrounded by water because it's too salty. So ocean water is too salty for people to drink. If you go to something like the Dead Sea, which is many, many times saltier, you would think nothing lives there. That's why it's called the Dead Sea. But no, if you get a microscope out, you'll find all these archaea and bacteria living in there. The halophiles, halo meaning salt, they love salt. They like environments that's much saltier than even the Dead Sea. So, you can put as much salt as you want in there, they like it. All right, let's now talk about, so we talked about heat, pressure, let's talk about acid. This critter here, I'm sorry, I'm choking on my words here. Picrophilus oshima thrives at a pH of 0.3 or 0.5 and down to a pH of 0. So what does this mean in normal terms? A pH of 0 is an incredibly strong acid, stronger than battery acid, stronger than sulfuric acid or any of those things. It would burn a hole in the table. This is really nasty stuff. Yet if you took that organism and just brought it up to a pH of 5, 7 is neutral. If you bring it up to a pH of 5, which would be slightly acidic, it would die because it's not acid enough. It really requires these things. The opposite extreme, alkali. There are organisms that can withstand incredibly high pHs. The equivalent would be things that you clean out your drain with, clogged pipes and clogged sinks. Very, very nasty stuff. You don't want to get it on your skin, but they survive there. And then drought. Deinococcus radiodurans can withstand being vacuum dried, not just dried, but vacuum dried, and survive for 6 weeks. Give it some water and it comes back. That's amazing. So some of these archaean bacteria derive their energy not from sunlight and photosynthesis like just about everything else does. When I was a kid 100 years ago, they told us the food chain ultimately goes back to the sun. The sun provides the energy for plants to do photosynthesis. They are the basis of all the food life on Earth. But that's wrong. Right? We know that's wrong. There are caves that are buried underground, completely sealed, and there are thriving colonies of bacteria and slime and stuff down there, and they don't care about sunlight. So we call these rock eaters, or lithotrophs. They self-generate their own food by eating rock. And so you can find them in caves, but you find them anywhere deep underground, like kilometers underground, not just a little bit, but many kilometers underground. You know, people drilling for petroleum or oil or those sorts of things, pull up the cores and, you know, they normally say, well, did we find what we wanted or not? And they throw it out. But other people say, well, let's look at it with a microscope and you find living organisms in there. It's amazing. So there can be this huge, huge biomass several kilometers below the surface of the Earth. Something to think about. In these caves, if you were to go into one of these caves, these little gooey things coming off the ceiling, they have this wonderful name called snotites. They're filled with sulfuric acid. You don't want to touch those nasty, nasty things. Okay, talking about extreme organisms at the bottom of the ocean, there's another energy source down there, hydrothermal vents, where you get heat from the mantle is kind of close to the crust of the Earth at the bottom of the ocean there, and it heats up the water to tremendously high temperatures. The water then comes up through these system of natural pipes, and it looks like smoke. They're called chimneys because it looks like there's smoke coming out, but it's actually just minerals dissolved in incredibly hot water. So you've got this rich source of minerals, which these archaea and bacteria can use as food. On this diagram here, I just want to point out off to your left, you got one of these black smoker chimneys, and if you read the temperatures there, inside the black smoker, right, the temperature is 375 Celsius. Just outside, it's 2, right? It's the bottom of the ocean, the temperature is 2 or 3 degrees. So there's this huge temperature gradient over a span of just a few meters, right? You got 2 degrees over here, 375 over here. You might be thinking, 375 degrees, shouldn't the water be boiling? Yes, if it was at the surface of the Earth, but we're deep, deep under the ocean. There's so much pressure, you would need much, much higher temperatures for water to boil. So it remains in a liquid state, even at 300, 400 degrees. So you've got this temperature gradient, you've got this hot water, which dissolves minerals and metals in it. It comes up into the cold water, it quickly cools off, precipitates out, and that's what the dark cloud of material is. It's metals and other minerals precipitating out of the hot water. Life uses that. It's a source of energy. Oops. Now, along with extremophiles, there's extreme adaptation. These are just, I'm going to give you a couple examples of critters that don't like extreme environments, but they can withstand it. So they're not technically extremophiles, they are extreme adaptations. So tardigrades are little microscopic animals, sometimes known as water bears or moss piglets. I like that name. They can withstand very, very high temperatures, very, very low temperatures, as long as you don't do it too quickly. You know, give them a few hours, they will adapt. Basically what they do is they shut down and go into hibernation. So this is not a good environment. Shut down, and when the conditions get better, they'll come back. They'll just wait. So they can take cold, they can take heat, they can take being zapped with X-rays. They've even done experiments where they brought them up into space, put them out, open the window, put them outside the space station, close the window, let them sit there for a few months, go collect them, bring them back. They're thriving again. Right. Now, do you know Coccus radiodurans? I mentioned this one, can withstand drought. Turns out it's one of the record holders for withstanding radiation. This is one of the ones that can withstand those gamma rays. It can withstand 3,000 times more than what would be lethal to a human. So you could just be, zapping this with X-rays and gamma rays all day long. Now, it's not that it doesn't do damage. It does. It destroys this little cell. It cracks the DNA and fragments it dozens of times, maybe even hundreds of times. It just chops the thing up, but it just repairs it. It's built mechanisms to repair the DNA. Why? There's no environment on Earth that has anywhere near this high radiation. So it's thought to be a repair mechanism for desiccation due to drought. When it's too dry and your proteins in your DNA fall apart. So you got to repair it. When the time gets good and the water comes back, you want to get rolling again. So it's got built-in repair mechanisms, and it can do this. More common bacteria, like the kinds we have in our mouth, if you give them a chance, they will and you don't change the conditions too drastically, you let it slowly change, they'll go into a hibernation state, build a little shell around themselves called a spore, and at that level, they're almost indestructible. I mean, you can put them in liquid nitrogen, you can put them in bleach, you can put them in hydrogen peroxide, you can heat them, you can dry them. They're basically indestructible. Give you an example of how indestructible they are. Amber is that gem, and sometimes inside the amber you can find little pieces of plants and animals. On the slide there you see a bee. Inside the bee's stomach were some pollen, and along with that pollen there were some bacteria. And people said, these are spores. I wonder, do they come back? And they do. Okay, so these are 40 million year old. So these bacteria just hibernated for 40 million years, came back as if nothing happened. Now I shouldn't say that. There is a big decline in the population. They all don't survive. But inside a bee's stomach there's probably tens of millions of these things. So if only one in 100,000 survives or something, you still have hundreds who make it. Here's another one. I just enjoy this because it's fun. Way back in the late 1960s, early 1970s, we had these Apollo missions to the moon. And before the astronauts went there, there was a big debate about what's the surface of the moon like. They knew it was very dusty. And one of the concerns is spacecraft lands and just goes 20 meters below the dust and just disappears. It's like, this is trouble. So they sent probes to land on the surface to see how hard it was. And the Ranger and Surveyor spacecraft are some of those that did that. So the Surveyor spacecraft went down and it landed at a particular site that they knew Apollo was going to, and they landed right next to it. So the Apollo 12 astronauts went out there and picked up pieces of the Surveyor, picked up the camera, in fact, and brought it back to Earth. Now, back in the lab, they took it and they swabbed it and put stuff on the Petri dish and stuff like that, and they were able to culture bacteria that came from that camera that was on the moon. So this is out in space, right? In the extreme cold, in vacuum, 20 degrees Kelvin, right? Minus 250 centigrade. And it was there for 2.5 years, and it just came back. So bacteria and archaea can survive these really, really harsh environments. This complicated diagram here, I just want to point out, in the center is life that we are familiar with. That's normal temperature, normal pH. But you can go way below the freezing point, way above the boiling point, incredibly low pH, incredibly high pH, and life thrives there. So this is new. These species were discovered in the last few decades. We had no idea how versatile life was until fairly recently. Okay. Finally, let's get on to part 3 because there's part 4 coming too. Part 3. So the topic here is the rapid origin of life on Earth. Now, we don't know how life started on Earth. That is a really difficult problem. Some smart guy said, we will never discover how life started on Earth. We need to invent it. Now, the problem is geology. Geology, plate tectonics have destroyed most of the rocks that go back 4 billion years and older than that. There just aren't many rocks around on Earth that you can do this with. There's some in Greenland, there's some in Australia in particular. If you go to those rocks, you can find things that look like microfossils. They look very much like a type of bacteria, cyanobacteria, photosynthetic bacteria. You can make all kinds of slides and they look like these wiggly little things here. And people say, oh, I know that species, I know that species. And then someone else comes along and says, that's just a wiggly thing in a rock. We see these all over the place. They're not formed by biology. So there is a controversy there. So those microfossils may or may not really be holding much evidence for the origin of life. One of the things that's not controversial, though, are these things called stromatolites. Stromatolites are these mounds made out of mud and silt and sand, and they're created by bacteria. Sticky bacteria. Bacteria like to live in colonies. They don't want to be, they're social creatures, actually. They want to live in colonies, and so they create this sticky mucilage, keeps them together. They make this bacterial mat. And it attracts the sand lands on it because of the currents and the turbulence in the ocean, land on the sticky stuff, and eventually it covers it up so that the bacteria have to migrate upwards above the sand. So you get these layered mounds. And they can grow for a long time. They are still here on Earth. They are still organisms alive, they're still living stromatolites, right? So the point is, we have stromatolites alive today. There they are. They're living in Australia. They like to live in salty water because it's too salty for most animals who like to eat them, right? There's a nice colony of bacteria. Let's eat it. Shrimp love these things. So if the water is very salty, it's less competition, or it's a safer environment for them. Anyway, you have fossils of cyanobacteria that go back 3.5 billion years. Right? And this is not controversial. And inside, you look at those fossils, and they may or may not have wiggly things in there, but the stromatolites themselves go back 3.5 billion years. The thing that pushes it back the furthest, and the most technical thing here, but I'll spend a minute on it. We've heard of carbon and carbon-14. Right? Carbon is the element of life. Carbon has radioactive states and other states which are not radioactive. Our normal carbon is called carbon-12. That's the 12 is the number of it's the atomic weight, number of protons and neutrons. You can have carbon-14, which has two extra neutrons, and it's unstable. It decays. So we can use carbon-14 for radioactive dating for things that go back, a few tens of thousands of years. But there's another carbon, carbon-13. So carbon-14 is good if you want to do radioactive dating, but after its half-life is relatively short, a few thousand years. So after 50,000 years, it's useless. So it's great for archaeology, but it's not good for geology. Carbon-13, though, is not radioactive. It lasts forever. So you can have life with carbon-12 and carbon-13. And in fact, we do. All of us here have carbon-12 and carbon-13 in our bodies. But biology, it's got to move a lot of molecules around. It's got machinery to do this. And it prefers the lighter version. Carbon-12 is a little bit lighter to move than carbon-13. So there's a preference to use carbon-12. So life does that. So when life uses carbon, it prefers to use carbon-12 instead of carbon-13. Not by a lot. About 25 parts per thousand, but it's measurable. And if you go look at the rocks on the Earth, and this complicated graph here, two things I want you to look at. If you look at that circled point there, that is rocks at the bottom of the ocean, right? It's just marine limestone. And you ask, what's the ratio of carbon-12 to carbon-13? You make that measurement. And we say, okay, let's define that as the 0 point. That's what geology does. Now let's go look at things that were made by biology, not to geology. And they're all shifted. They all have less carbon-13. So you can tell whether some substance was made by geology or biology by measuring the ratio of carbon-12 to carbon-13. So you do that, and it's pretty clear stuff on Earth is shifted by 25 parts per million. Then you go look at some of these Precambrian things. This is ancient material. Hard to find, but you get it, and lo and behold, you find that same 25 parts per thousand shift. This material was made biologically, not geologically. That's the interpretation. So we have this carbon-12, carbon-13 isotopic fractionation is the fancy term for it. The punchline is, though, we can tell that there is material that seems to have been created through biological mechanisms, not geologic or chemistry. This figure here, the x-axis, the bottom here, is time going back in the past. The present day is 0 on the left. As we go back in the past, we go towards the right. 0 up top, these white circles, that's rock that was made through geochemical processes. That's our 0 point. What we have below it, at about minus 25 parts per thousand, are all these samples from different ages rocks going all the way back to 3.5 billion years ago. The oldest ones go to 3.85 billion years ago. So 3.85 billion years ago, we have evidence that there was life on Earth. And it's evidence that implies not only life, but pretty complicated life. This carbon isotope thing usually is one of the things that comes from photosynthesis. So it shows that life was there at least 3.85 billion years ago. Now, why is that significant? So let's recap what we did here. We've got significant life going at least 3.5 billion years ago from stromatolites, and this and some controversial evidence from microfossils. The carbon-12, carbon-13 isotope ratio is not as direct a probe of whether something was originated from a biological system, but it's pretty good. And that goes back 3.85 billion years ago. Now, why is this number important? The Earth is 4.5 billion years old. 3.85, that's a long time between 4.5 and 3.85. But the astronomy tells us, especially samples from the moon, that the inner solar system was bombarded by giant asteroids and meteors and all this stuff up until about 3.9 billion years ago. So this is known as the Great Heavy Bombardment, the Great Late Heavy Bombardment. And the Earth was pelted with these things, Mars, the moon, hit with giant asteroids, not little ones, I mean big ones frequently. And they would hit so big they would vaporize the oceans. So this went on up until about 3.9 billion years ago. So now the clock doesn't start at 4.5 billion years ago. It really starts at 3.9. And at 3.85, we have evidence of life. So it seems that as soon as the conditions are right, meaning no asteroids are coming in and vaporizing the surface of the Earth, as soon as the conditions are right, we have evidence of life. It did not take billions of years. It barely took 100 million years. Well, that sounds like a long time, but geologically speaking, that's very fast. So it could have been that life started and was obliterated many times between 4.2 and 3.8 billion years ago. We don't know. All we know is something survived, and we've descended from that. The implication of this 3.85 billion years ago is that it implies that if the conditions are right, life will arise, right? It's not a one-time miraculous event that takes billions of years. All right, I'm running late, so I will go through this quickly, but you can't miss this part. All right, in the outer part of the solar system, we have the icy moons. Here's a sketch of, or actually photographs of these different objects out there. We got Earth and Venus and Mars. Those are the terrestrial planets, and Mercury's in there, shifted over a little bit. In between Mercury and Mars, we've got two moons, Ganymede and Titan. There are moons in the solar system bigger than planets. Way off to the right is Pluto, which we're not even going to talk about Pluto. There are a lot of big objects in the outer solar system that if they were going around the sun by themselves, we just call them planets. Here's another view of some of these. Jupiter has 4 big moons discovered by Galileo. And going from the inner part to out, we got Io, Europa, Ganymede, and Callisto. Why are these 4 important? Well, this slide is kind of giving away what's happened. Their orbits are not perfectly circular. And because they're not perfectly circular, there are tidal forces. So on Earth, we have these tides, right? The moon causes the tides. We get these tides every day. Imagine if you replace the moon with Jupiter, right? 315 times, 312 times the mass of the Earth. The tides would be enormous. And so you wouldn't just move the oceans. You'd move continents up and down, right? And you'd be really stretching and compressing the planet as it went around. So this is what happens. So this is Io. And it's the closest moon, closest of these 4 big moons to Jupiter, and it's basically molten. We're way out far from the sun. It should be completely cold out there. It's incredibly cold. We expected everything to be covered in ice. This planet's covered in lava. Right? And and you can see that thing up there is a plume from a volcano. And for the scale, look at the size of the object and the size of that volcano. I mean, the stuff is being squirted out into space. There's a picture of the night side, a thermal camera picture. You can see the hot spots of the volcanoes going off. So that's the innermost moon. It's so hot, the rock becomes magma. If we move out, the tidal forces are less. We get to Europa. We expect a lot of ice out there, and there is, but there are also tidal forces. And because of the tidal forces, you don't melt the rock, you melt the ice. If you melt the ice, you get water. That's the interesting. Underneath the crust. So the crust is very smooth, hardly any craters, which tells you it's very young. It's got all these cracks on it, and these cracks are from the tidal forces of Jupiter compressing and stretching. these beautiful cracks, and they come and go. Right? They're like ice on ice rafts on the sea. They move and look like a jigsaw puzzle up here. Underneath the crust, there's pretty good evidence that there's liquid water. I won't go into the evidence. You just trust me. There's evidence. We don't know how deep. That's the hard part. We don't know how deep below the crust, but there's a good amount of evidence saying that there's a lot of water underneath the crust. And when I say a lot, how much? Well, before I go there, you've got liquid water. You've got a source of energy, this tidal energy, not sunlight. You've got tidal energy, and you've got organic material, carbon molecules. Carbon molecules are everywhere, one of the most abundant elements in the universe. So you've got the three ingredients you need for life. So this picture here, if you took all the water on the Earth and put it into a little sphere, it's actually not a lot. We think of the Earth as having a lot of water on it, but it's a lot of water on the surface. As you go deep down, there's not as much water. So you put all the water on Earth together, and it's actually a small amount. You compare that with all the water on Europa, liquid water. Europa has more water, liquid water, than the Earth does. So that's significant. Moving outwards, we got Ganymede and Callisto. They're bigger moons. They're further out. They have less tidal forces. There's some evidence that maybe water on Ganymede. Callisto probably is out of luck. All right. If we go to the next planet out, Saturn. Saturn's got a moon, Titan. Titan is a very, very big moon, and it's got a dense atmosphere. There's a picture of Titan, and you see this orange stuff. Why is it orange? Because it's got a thick atmosphere. Pause for a second. We have a moon with a thick atmosphere. It's got air, but it's really cold. That's the problem with Titan. It's 85 degrees Kelvin. It's minus 180, 190 degrees Celsius. But it's got air, it has clouds, it has rain, it has rivers, it has seas, it's got mountains, all that good stuff. Thick atmosphere. In fact, it's got more air than the Earth does. But it's mostly nitrogen, and then a lot of organic material, ethane, propane, that sort of stuff, hydrocarbons. So we can't see the surface because of all this hydrocarbon smog in the atmosphere. But we do know there are clouds of nitrogen. It rains liquid nitrogen, if you think about it. It seems like science fiction. It rains liquid nitrogen. There's lakes and seas of ethane and methane and things like that. I would love to have a barbecue there, a braai there, because you have to bring a tank not of propane, but of oxygen. Okay? The atmosphere is already filled with propane, but you need the oxygen. So you bring the oxygen tank, you hook it up, you start cooking things. So when you cook, you produce, when you burn these hydrocarbons, you make carbon dioxide and water. And if you think about it, it will go up in the air, but it's really cold, so very quickly it will condense, and you'll form clouds, and it'll start to snow. So right next to your braai, you'll be cooking up, and it'll be snowing all around you because you're making your own weather right there. So I would love to do that. So Titan is filled with organic material. Organic material is building blocks of life. It's got atmosphere, it's got liquids. It's just cold. So it won't have water like life on Earth because we require liquid water. It's much too cold. Water is harder than rock on Titan. But it's an interesting potential. If there was life on Titan, it would be super, super interesting because it would not be like life on Mars. It would be much more interesting. One last stop before we go. Enceladus is a moon. It's a little tiny pipsqueak of a moon. It's going around Saturn. Nobody thought too much of it. It's a little weird. It's kind of bright on one side, dark on the other. It's small. It's a really small moon. Everyone says it's a snowball, just like everyone else out there. But if you look at it, you don't see craters on it. You see these cracks. These cracks are just like Europa. It suggests that maybe there's liquid water under the surface. How can that be? We're much further from the sun than Jupiter is. The tidal forces are much less. There's no way there should be liquid water on this little tiny rock. It's a puzzle. So what created these cracks? Don't know. But it does look suspiciously like Europa. And then we got this image here. Right? This is looking more towards the sun, and you see a crescent Europa. And you see this fuzzy stuff on the bottom. You say, oh, something wrong with the camera. And so you enhance the image. You see more fuzzy stuff. It's like, that's weird. What is that? And you go back and you say, well, let's look at other images. And you say, this sometimes is there, sometimes it's not. what's going on? And then, you put it together and you start looking at it more carefully. You go, oh, these are geysers erupting on the surface of Enceladus. Right? So there is water being squirted up into space from this moon here. Why? I mean, it's this little cold rock. Well, no, it's not cold. It's really hot on the inside, and it's got this geologic activity enough to shoot water up into space. So there's quite a few of these. So here's an object in the solar system that we know of that has water on it, liquid water. And we're not just speculating that maybe under the crust. No, it's shooting out into space. We have spacecraft that have flown through it and measured the water content and the molecules and stuff in the water. So we have a place in the solar system right now that has liquid water, and it's kind enough to spit it out into space for us. So you've got liquid water. There's the composition of that water suggests that it could be minerals and hydrothermal vents and stuff like that. It could be an even better place to find life than Europa. But it's a small place. Okay, so now let me finally wrap it up. So there have been 4 major discoveries. We got these exoplanets, which now tells us we have billions of worlds to explore. This was not known about 20, 30 years ago. Changes the whole picture. We've got extremophiles, which have extended the limits of life tremendously. Things can live where we never imagined could have lived before. That's also changed things. We've got a very quick origin of life on Earth. It didn't take billions of years. It was, geologically speaking, instantaneous. Now, that's controversial, but what's not controversial is it did not take billions of years. Right? Whether it took a few 100 million, 300 million, well, those are details. Right? It was quick. Right? So that means if it happens that quick, it can't be that hard to do. And if it's not that hard to do and it happened on Earth, well, it probably could happen somewhere else. One of these other billions of planets where these extremophiles can survive. And then finally, in our own solar system, we've got at least 3 places that life could be thriving right now, unlike Mars. Mars is dry. Maybe it had life in the past, but not now. But right now, there are 3 places which could be hosting life, but none of those 3 places are in the habitable zone. So don't limit yourself to the habitable zone. You can go much further out. There's other sources of energy. So with these 4 topics, I think, thank you. Thank you. I think the study of astrobiology has kind of reached the next level where we really have much better data and a much better clue and a much deeper understanding of how likely it would be to find life in the universe. So with that, thank you for your attention.
So, at least now I'm sure everyone is awake, but I'm also very sure no one would have fallen. No one would have fallen asleep. No, thank you so much, Professor William Welsh from San Diego State University. This was a magnificent talk, and you a couple of different, all very interesting topics. I'm pretty sure there are some questions from the audience. We start at the back.
Thank you for that engaging talk. What would you and some of your colleagues at the Kepler, what do you guys think of panspermia, extraterrestrial origins of life, sort of cosmic contamination? Is there any credence to that?
Right, so for the people online, the question was, what do astronomers and people on the Kepler mission think about the idea of panspermia? Panspermia is the idea that life came to Earth from somewhere else in the universe. Spores maybe landed on the Earth and life started that way. The problem with that hypothesis is not that it's wrong, right? You can have spores travel from Mars to the Earth. You can have spores from Earth travel to Mars. So if we found life on Mars and it's the same as life on Earth, it's not so shocking because it could be the same. Right? So panspermia is not a crazy idea. The problem is it doesn't tell you anything. It just said life didn't start on Earth. It started somewhere else. Well, where else? And how? We don't learn anything from that hypothesis. It's kind of a sterile hypothesis in that, yeah, it could happen, but it doesn't tell us anything about how life originated elsewhere. And so it's not a very useful hypothesis. It's less likely than just having life originate on Earth because we know from Miller-Urey experiments, a whole bunch of other things, creating complex organic molecules is simple. You can create oceans of complex organic molecules just without, if the atmospheric conditions are right, you can do that without much trouble. So you really don't need to bring in life from somewhere else. Now we don't know that because we haven't invented life yet. But probably panspermia is not necessary. One, it's not necessary, and two, it doesn't tell you anything. That's the problem.
Further questions?
Thanks for the talk. I much enjoyed that. Linked a little bit to the last question, maybe even a fifth point in your presentation there. What about the development of organic chemistry in space? We see we picked up amino acids, for example, in space, things like that.
I'm sorry. Could you repeat the question part?
It was a little bit of an invitation to discuss. We see things as complex as amino acids in space. It's maybe not quite panspermia, but those could very easily have been the starting blocks why life started so early on the Earth
Right.
and other places.
Yeah, so very, very good comment. The comment was that if you look at material in space, meteorites, for example, or just interstellar dust and gas, you find organic material, and not just carbon, but biocarbon molecules, things like amino acids, sugars. We found DNA nucleotides, right? You find all these kinds of things, they just naturally form. So when meteorites land to Earth, there are a couple of different types of meteorites. We just saw some meteorites today. Those are the iron meteorites downtown. They're not the kind that would bring life to Earth. There's another type there called the stony type. And of the stony type, there are carbonaceous chondrites. They're made mostly of carbon. So you've got asteroids that are made mostly of carbon. You analyze that carbon material, and it's got things like sugars and amino acids and the building blocks of DNA right in there. So the materials you need for life to begin are on meteorites. You just come to Earth, right? You put them in a nice warm soup, give them enough time, maybe something interesting will happen. Now, there is one important thing, though. When you look at these specimens from space, they have equal left-handed and right-handed varieties. Molecules can have left or right-handedness. They're mirror images of each other, like your left and right hand. They are the same weight, the same chemical composition. They work the same way, but they're not identical. They are mirror images. Your glove for your right hand will not fit on your left hand. So these molecules don't work. Biology on Earth is extremely specific about handedness. All the amino acids are all left-handed. If you have a right-handed amino acid, useless. Similar thing with sugars. By the way, artificial sweeteners are the wrong-handed sugar, right? They can taste like sugar, but you can't get any nutrition out of them. So that's one use or misuse of this. They're called chiral molecules or enantiomers. In any case, you got this handedness. Biology produces one hand, always uses one. We don't know why it chooses left hand. It could have been a flip of a coin. Once one hand starts to win and you got like 55% versus 45%, it runs away to 100% because all the molecules you produce are left-handed. And everything anyone eats is left-handed and will produce as left-handed. So if there's a little extra left-handedness, it will go all the way because if you're a poor right-handed bacteria, you got nothing to eat. Right? Everything is left-handed. So you become one-handed. So biology on Earth is one-handed. The material you get from meteorites is left and right. So it's both. So we know that the organic molecules in meteorites are created by chemical processes, not biological. So that's one of the ways you know. If you get a meteorite from space and you check the molecules in it, if there's a specific handedness, you say, oh, that's been contaminated by stuff on Earth. If it's equal left and right, or close to being equal left and right, then you know it's really pristine material and it's from space. But great question. Thank you for that.
First of all, for a great presentation. I just wanted to ask you a bit of a question. In terms of your list, or what's the next interesting space mission or thing, that tool you would like to see, or what's out there that you're most interested in at the moment?
Great question. So we're almost there, right? Ever since I learned about Europa and the potential of vast oceans of water under the shell of Europa, I was like, it's so close, let's just go there. We sent spacecraft past, way past Jupiter. We can do that. We have the technology to go to Jupiter. Let's do it. And well, it just costs money. So I've been hoping for a mission like that for like, well, 20 years now, over 20 years. I'm thinking, oh my God, I hope something happens before I die, but something has happened. There's 2 missions on their way to the icy moons. One has been launched by the European Space Agency. It's going to Ganymede. The other one is called Europa Clipper. It'll be launched, I think, later this fall, and it's going to Europa. Now, it's not going to land and drill down. So the wishful thinking, I've seen the cartoons or the schematics for this. The lander lands down, and it got to drill through the ice. Well, you think about it. That's hard to do. How do you drill through the ice if you don't have any equipment? You don't have a drill. Right? You're not bringing a drill with you and all the rigging material and all that stuff to Europa. You don't have the money. You don't have the technology to bring heavy stuff. So how do you get through the ice? Well, one idea is you just make it really hot, and it'll just melt through the ice by itself. It would work, and you just get it hot enough. Getting things hot is not hard. And you just go all the way down right until you get underneath the icy level, and you get into that ocean. Well, it's easier said than done. So the Europa Clipper is not the mission to do that. It's going to go to Europa and study the moon, Europa, very carefully to learn where is the best place to put the lander. Because you get one chance to do it. It will take like 12 years of work to do anything, and it may take years of melting through the ice. You got to get it right. And if you imagine you're melting through the ice and you don't have enough, and you stop a kilometer short. Oh, what a disaster that is. Or you're coming down and you hit a big rock. Trouble, right? So the Europa Clipper has radar that's ice penetrating. It will find the best place to go, and you should be able to melt your way through the surface. So a mission to Europa would be highest on my wish list. And while we're not going to have a lander in the next decade, at least we'll have a probe out there to tell us where a lander should go. And so there's hope that that will happen. Yeah, it's cool. It's called a cryobot, not a robot, but a ice. It's an ice robot that has to melt through the ice. So they actually practice. I don't know if you know, there's a place in Antarctica called Lake Vostok. There are these oceans, well, seas or lakes. They're lakes. They're lakes deep under the ice, kilometers under the ice. And they've been isolated from the surface of the Earth for millions of years. And so you can practice on Europa by trying to go down into those lakes down there. Now there they cheat, they just drill. But you could practice and actually do it. And you're finding there's whole fascinating ecosystems down there that are distinct from life on the surface. Some things are the same. You go down and you see shrimp down there. You're like, what the? There's shrimp down there. There's shrimp everywhere. But you find bacteria, microbes, and there's shrimp who want to eat them. So we go to Europa, you'll find shrimp, probably the first thing. You could, it sounds like it's so far away you couldn't do photosynthesis, but you could. There are organisms that specifically deal with very low light levels, and they can do photosynthesis near the bottom of the ocean. Like, there's not a single photon from the sun that can get down a kilometer to the bottom of the ocean. It's pitch black. Well, you go down there, it's not pitch black. Why not? Because you got these hydrothermal vents, and they've got so much energy. It's not thermal energy. It's not glowing from thermal energy, but it produces enough that you can produce. There are chemical reactions that can create a little bit of light down there. And then there's bioluminescence itself. So there's enough light down there to do photosynthesis at the bottom of the ocean. That's much more light than you would get at Europa, even under the ice. So there's probably hydrothermal vents on Europa. I'm not worried about it. Well, the reason I mentioned photosynthesis is because it produces oxygen. Now, oxygen for us is a good thing. For most life, it's a terrible thing. Right? You're like, what? You think about it, when you go to the health food store, they always talk about antioxidants. Why? Because oxygen is poisonous. Oxygen will kill you if you have too much of it. It's really, it's extremely reactive. So oxygen radicals will just destroy your cells. So you got to be very careful, and you got to be able to, your body has to be able to repair itself or make itself immune from these oxygen radicals. Vitamin C, one of the things that help. But oxygen has a great potential. It gives you so much energy, right? It's a very reactive molecule. If you can tap into that, you've got 30 times more energy than if you don't tap into oxygen. So oxygen is not required for life, but it sure gives you an energy boost. So if there's photosynthesis, then you have the oxygen probably.
All right. With the energy boost, I think we start thinking about energy boosts for ourselves from the very back of the hall. So let's thank Professor Welsh again very warmly. So let me leave with two more things. Well, the one is we obviously can stay in here for quite a while. Please just help a bit to put the chairs to the side, and then we can stay here, mingle, and have all these interesting chats. The other question is, you might have asked yourself, is how come someone from San Diego State University will be visiting Namibia and us here? And this is obviously for two reasons. Well, the first reason is that Namibia is just a great place to visit, right? But there's a tiny addition to that, and that's just in 2 weeks' time, there is the International Astronomical Union's General Assembly. So that's a meeting of professional astronomers. This happens every third year. And this year, it will be in Cape Town, and with that, it will be the very first time in its history to be on African soil, and we're all very excited for that. So there will be also a group from Namibia going there, and even a whole booth, a Namibian house, so to say, will be hosted at the General Assembly, bringing together all the different stakeholders, be it the colleagues from NAS, be it the colleagues from UNAM, be it the Scientific Society, the National Commission, and yes, so we're all very excited. Thanks again, Bill, and I will hand over to Ruth for the final words, for closure.
Going to be very, very, very sweet, short, and final. So I hope with all the life that is possible that we will have some visitors at some stage. My father always said, if this UFO lands on our farm, in front of our farmhouse, the first thing I'm going to do is offer him a whiskey. Unfortunately, my father passed away, so we were never able to offer a whiskey. The closest we got was a clippy key that came from out of space, apparently, and blew our water basin apart. So that was the closest encounter we had with out of space. Thank you very, very much. I hope we see you again here soon with new developments. As for the future, as you might have seen, we only have the AGM coming up. The tour, unfortunately, is fully booked. And then at the AGM, yes, of course, it's going to be the last AGM before we turn 100 next year. And yes, I was told, unfortunately, we had a bit of technical difficulties, as you might have noticed. So usually I would promise you now that the video will be online in a couple of days. We're going to try and make a video out of this. I have already been informed that we are allowed to keep the PowerPoint presentation, so would you like to have a copy, please contact us. And we will try to put it on YouTube as per our regular videos. And with that, I hope to see you all at the bar. Thank you very much.